using a pipette for a dilution, it must first be primed with the solution being measured. Start by cleaning the outside of the pipette to prevent contamination. Then draw a small amount of the stock solution into the pipette and release it back into the container. This coats the inside of the pipette with the solution and helps ensure more accurate measurements during the dilution process. The dilution equation used to determine the volumes for each dilution is M1V1 is equal to M2V2. In this equation, M1 represents the concentration of the stock solution, and V1 is the volume of that stock solution. M2 is the desired concentration of the diluted solution, and V2 is the final total volume after dilution. By rearranging this equation, we can calculate how much of the stock solution or dilutions is needed to reach our next target concentration. To prepare our first dilution, we transferred 12.5 milliliters of the stock solution into a 25 milliliter volumetric flask using a pipette. This measured volume represented half of the final solution volume. Next, we slowly added distilled water to the flask until the total volume reached 25 milliliters. While adding the water, we carefully observed the meniscus and adjusted the level so that the bottom of the meniscus aligned exactly with the 25 milliliter calibration line on the flask. Ensuring the meniscus was level with the mark helped us maintain an accurate final volume and produce a precise dilution. A meniscus is the curved surface that a liquid forms when it touches the sides of a container, such as a graduated cylinder or a volumetric flask. This curve occurs because of the attraction between the liquid and the glass. When measuring liquid volume, it is important to read the bottom of the meniscus at eye level to avoid measurement errors caused by parallax. Properly aligning the bottom of the meniscus with the calibration mark ensures that the correct volume is measured. This is especially important in dilution experiments where small measurement errors can affect the final concentration of the solution. For our second dilution, we transferred 2.5 milliliters of our first dilution into a 25 milliliter volumetric flask using a pipette. After adding this measured volume, we slowly filled the flask with distilled water until the total volume reached the 25 milliliter calibration mark. While filling, we carefully watched the meniscus and made sure the bottom of the curve aligned exactly with the calibration line. This ensured that the final volume was measured accurately and that the dilution had the correct concentration. For our third and final dilution, we transferred 2.5 milliliters of the second dilution into a 25 milliliter volumetric flask using a pipette. After adding this measured amount, we slowly added distilled water to the flask until the total volume reached the 25 milliliter calibration mark. While filling the flask, we carefully observed the meniscus and ensured that the bottom of the meniscus aligned exactly with the 25 milliliter line. This step was important to make sure the final volume was measured accurately and that the dilution had the correct concentration. Once all of our dilutions were completed, we had a series of solutions with decreasing concentrations. The original stock solution had a concentration of 0.02 molarity. Our first dilution had a concentration of 0.01 molarity. The second dilution had a concentration of 0.001 molarity, and the third and final dilution had a concentration of 0.0001 molarity. These stepwise dilutions allowed us to create a range of known concentrations that could be used for further analysis.